The next part in that lesson is packaging of DNA. That is related to the length of the DNA, how far it is organized inside the nucleus, and what is the arrangement, etc. Now, if we are taking the length of DNA, the length of DNA is expressed as the number of nucleotides in terms of nucleotides or nucleotide base pairs, simply called as BP. Now, the number of nucleotides and or base pair is constant for a particular individual. I am giving 4 examples. In the case of pi into 174, bacteriophage, the number of nucleotides is about 5386. This is the number of nucleotides. If we are taking what's called the lambda phage, bacteriophage, another one, the number is still higher, 48,502 base pairs and nucleotides. If you are taking this E. coli bacterium, I am taking here the third one, E. coli. So it has 4.6 in 10 power 6 nucleotides and base pairs. We can express either nucleotides or nucleotide pairs or in the form of base pairs. 4.6 10 power 6 base pairs in the case of E. coli bacterium. In the case of human beings, we have 6.6 into 10 power 9 base pairs. This is in the case of diploid cell. In the case of diploid cell, we have 6.6 into 10 power 9 base pairs. Then what is the number of nucleotides present in a haploid cell? So in the case of haploid cell, see in human cell, we have 3.3 into 10 power 9 base pairs. So this row, actually the base pairs are the nucleotides found in different organisms, but it is constant to a particular organism. So an example of pi into 174 bacteriophage, 5386, then lambda phage, 48,502 nucleotides. In the case of E. coli bacterium, we have 4.6 10 power 6. In the case of human diploid cell, 6.6 into 10 power 9, or in the case of haploid cell of human, 3.3 into 10 power 9 base pairs. Length of DNA and the number of nucleotides or base pairs is constant for a particular species. For calculating the length of the DNA, we need 2 criteria. Number 1, that is number of base pairs. This is number 1, number of nucleotides or number of nucleotide pairs. The second one, that is distance between consecutive base pairs. These are the 2 criteria needed for calculating, that is the length of the DNA. If you are taking DNA, it is a double helical structure. We have seen already. This is a double helical structure. We have 5 dash, 3 dash, 3 dash and 5 dash. If we are taking what we have, the diameter, it's about, you know that one, 20 angstrom in diameter or 2 nanometer. Now we have many turns in the nucleic acid, many turns in a nucleic acid. This is 1 turn, or we can say 1 pitch. Now the distance or length of 1 turn is equal to 34 nanometer, or I made a mistake, there is 3.4 nanometer or 34 angstrom unit. Now this is 3.4 nanometer. Now how many nucleotides are present per turn? So number of nucleotides per turn is equal to number of nucleotides per turn. This is equal to 10. So now we have the distance between consecutive base pairs. What is the distance between consecutive base pairs? The total length is 3.4 nanometer. It is being divided by 10. So we are getting the value 0.34 nanometer. This is actually distance between consecutive base pairs or distance between the adjacent base pairs and nucleotides. That is what is called 0.34. I mentioned we need number of base pairs. That is one criteria. Another one, the distance between consecutive base pair, that is 0.34 nanometer, is constant for all the DNA except ZDNA. In the case of ZDNA, its value is about actually 0.375 nanometer. This is in ZDNA. All other DNAs have the equidistance or distance between the consecutive base pairs is always 0.34 nanometer. Now, so we need number of base pairs, the distance between consecutive base pairs we already calculated. These are the two criteria to calculate what is called actually the length of DNA. Now I'll take the length of DNA, the length of double helix DNA in, that is human or mammalian cell. If you want to calculate the length of DNA double helix in any cell, now the total number of base pairs in that cell or in a species is multiplied with the distance between consecutive base pairs. We can take either, for example, total number of base pairs, for example, in the case of human being, 6.6 into 10 power 9. If you want to calculate the length of DNA in the case of E. coli bacterium, 4.6 into 10 power 6. So we can calculate for any species. So this is constant for a particular species, that should be taken. The distance between consecutive base pairs, it is same in all the just organisms excepting the ZDNA. So it is the distance between consecutive base pairs is constant in the case of ADNA, that is BDNA, CD and eDNA, but that is in the case of ZDNA, the distance is different. For example, the total number of base pairs we have to calculate in a mammalian cell. It is about, you see, 6.6, 10 power 9. This is the total number of base pairs from which we can calculate the length. Now the equidistance is about, you know that one, that is 0.34 nanometer. If it is ZDNA, it is equal to, you know that one, just in the case of ZDNA. Now the distance between the base pairs is about 0.375 nanometer. So according to the nature of the DNA, we can just use this one. It's constant for all the DNAs except ZDNA. Now if you want to calculate, that is what we have, the length of the DNA in meter, this value must be multiplied by 10 power 9, 10 power minus 9 meter base pairs. If you want to calculate the length of the DNA in terms of centimeter, then it should be multiplied by 10 power minus 7, that is centimeter per base pair. Or if you want to calculate in terms of what is called millimeter, 10 power minus 6 millimeter per base pairs. So according to the unit value, whether it is meter or centimeter or millimeter, we can just multiply and get the answer for that one. Now here I am calculating, there is a length of the DNA in terms of meter. Okay, I am taking that one, the value, that is 10 power minus 9 meter per base pairs. Now if it is a value, just I am writing here 10 power minus 9, that is meter per base pairs. So it is approximately equal to 2.2 meter. This is the value what we are getting. So the length of the DNA in a mammalian cell is far more, that is greater than what we have the dimension of the nucleus. So that the DNA must be folded to fit into the nucleus. That is one. Okay, we have another problem. Suppose the length is given. How can you calculate the total number of base pairs? So I will give another problem. This is one. How to calculate the length of the DNA in terms of meter. So 2.2 meter base pairs. In terms of length, 2.2 just. So I am not writing the base pair. This is only the value, the length only I am giving, not the number. So 2.2 meter. Suppose if the total length of base pair is asked. For example, in one problem, the length of DNA in E. coli, the value is given 1.36 nanometer. It was given the length of DNA in E. coli. They are asking about calculate the total length. Calculate the total number of base pairs, not the length. The length is already given. You have to calculate the total number of base pairs. The question is how to calculate the number of base pairs. Okay, so now I am taking simply length of DNA double helix is given. So I am taking the length. We have to calculate the value. So length is given, that is about what we have, 1.36 nanometer. So if you want to represent the value in meter, it should be converted into meter. 1.36 actually minus, that is 3 meter. I am converting this value into meter first. So here, so we have to calculate this one. The distance between these two, the distance between two successive nucleotides or consecutive nucleotides or bases will be 0.34 nanometer. So we have to calculate in terms of unit meter. So 10 power minus 9 meter base pairs. So this is the one we have to calculate. Now what is the answer for that one? So we have 1.36, that is minus 3, that is meter. Actually this is 1.36 minus power meter, and we have to represent this one 0.34, the value. We are bringing all these things in this one to calculate what we have, the number of base pairs. Calculate the number of base pairs. So 1.36 minus, I'm writing in this way, minus 3 meter into 10 power minus 3, that is meter. I am not writing that value. So 1.36, so I am converting this one into meter. So this one, now 0.34, the value. Then we have 10 power 9. We are bringing this sign. Now what we are getting actually, see, this is 4. So now 4 into, that is power 9 minus 3, we have 10 power 6. Such a huge number of base pairs in, that is per meter. Number of base pairs per meter. So meter per base pair or number of bases, these are all the number of bases per meter. So this is the value you can calculate. So on both the sides we are calculating by using what is the meter value, the unit. That's why this is the answer 4 into 10 power 6 base pairs per meter. So this is about another calculation. If the value is given regarding the length of the DNA, you can calculate the number of bases also, because you know they won't give in the question paper, because the distance is always constant. The number of base pairs in a particular cell is constant. So you have to calculate the number of base pairs, or you can calculate the length as given in the question paper. This is the answer for that one. Okay. The chromosomes are the carriers of genes which are responsible for the transmission of characters. A person by name Duprat in 1965 given a single stranded model for DNA. Single stranded model for DNA. That is called unimeme. If it is single stranded, it is called unimeme. Okay, single stranded model for what is called the DNA, along with a coiled molecule associated with proteins, named the histone. Simply he explained. That is, the DNA is a single stranded molecule, a coiled molecule associated with the proteins. This is the one what is given. Normally, if you are considering the amount of DNA, the amount of DNA is more in the case of animal and plant cells when compared to, that is, the prokaryotes and bacteria. So, the large DNA, a long chain of DNA, must be normally put into the cell and packed into the cell. That is what we have seen not in the case of dimension. The mammalian cell, a long chain of nucleic acid, far greater than the dimension of the nucleus. A nucleus having a dimension of 10 power minus 6 meter. This is the dimension of the nucleus. Such a dimension of nucleus has normally the number of base pairs to 0.2 meters. So it must be packed into the cell, further it should be folded. Now in the case of prokaryotes, though a definite nucleus is absent, we have in the case of bacteria, that is, the nucleus is called nucleoid. There is no definite nucleus. Though definite nucleus is absent in the case of prokaryotes, it is being represented as nuclei. The DNA is not scattered in the cytoplasm. Now, the DNA is normally positive or negatively charged, is being held in a region, with or provided with what's called positively charged proteins. So the DNA is held in a region, it's not being scattered in the cytoplasm, it is held in a region where you have positively charged protein responsible for holding the nucleic acid DNA in that region. That region is called nucleoid. In the nucleoid, the DNA is organized in the form of large loops held by proteins. The nucleoid contains the DNA organized into large loops held by protein. So in the case of prokaryotes, normally there is no definite actual nucleus. The DNA is circular. And lacking chromatin organization. There is no formation of chromatin organization. The meaning for that one, the DNA is not associated with the protein to form a chromatin fiber. So it is lacking chromatin organization. Such a DNA of prokaryotes, which is circular and lacking the chromatin organization, is called genopore. Now this is the bacterial cell showing what is called the nucleoid, genopore and bacterial cell. Now nucleoid, a region where the negatively charged DNA is held by positively charged proteins. That is the one. The positive nature of the protein is because of the presence of amino acids, basic amino acids like arginine and lysine in their side chains. So that is because of that one, the amino acids arginine, lysine, the proteins are positively charged. So it holds the DNA in that region. The region where the negatively charged DNA is held by the positively charged protein is called what is known as nuclei. Now in that region, the DNA forms large loops held by protein. Now in prokaryotes like bacteria, the DNA is circular without what is called chromatin organization. So such a DNA which is circular without what we have the chromatin organization is called genopore. Similarly, we can say the bacterial DNA is called genopore. Circular, no chromatin organization. There is no formation of chromatin. So that's why it's called as a genopore.